Okay. Okay. Allele. Remember allele. Version of a gene. We say the allele you got from your mom, or the allele you got from your dad. Okay. Which of the patterns can't belong to a child of this couple? Okay, so here's mom, and here's dad. Okay, so A could be, and so this one could be a kid, absolutely. This is dad's allele, and this is mom's allele, so that's good. This is mom's allele, and this is also mom's allele, so this can't be it. Okay? And then this can be mom and dad. Mom and dad. Okay? Which cannot be the child of this couple? Huh? C? Now remember one thing here. This right here is an example of a double band. Okay? So, in other words, mom has it and dad has it. So that could definitely be both. Okay? So that's okay. A is, this is dad, and this is mom. And then this is dad, and this is mom. And then this is a double band too, but mom doesn't have one, so this cannot be it. Okay. What kind of cell is this? Right. Okay. Why do we know that? What do we have here? We have a nucleus. Okay? Bacteria or archaea do not have a nucleus, nor do they have these things called mitochondria or any of the other stuff. They just have DNA inside of a cell, no nucleus or anything like that. So only a eukaryotic cell has all this other stuff. So who are these guys? A, B, or C? Oh, that's not right. Yeah, okay. B, C. Okay, now what? B. Okay. Yep, Watson and Crick. Sorry about that, I'll have to fix that. And the reason they figured it out actually was because they took, you guys haven't ever taken chemistry, maybe in high school. Well, sometimes you get these different atoms and you actually put them together into a molecule. Well, they pretty much did it that way. And it helped them figure out where things had to go. Okay. Um, so, what goes here? What goes here? Okay, good. Okay, so what we have is we have a double-stranded helix, remember? So, double-stranded, twisted. When there's replication of the cell, you have to replicate all the DNA. So what you do is, all this DNA unwinds, and it then adds the complementary strand with something called DNA polymerase. An investigator finds a small amount of tissue at a crime scene. What procedure will they use to make the small amount into a large amount? PCR, not STR. Okay. STRs are highly repetitive variable. In other words, they're variable, and there's a lot of repeats in them, and they vary among people. Okay, so short tandem repeats. They use about 16 or 17 or more now. Almost no other person will share the same length of the repeats at each of those specific sites. And then to actually separate the short tandem repeats, whether they're 2 repeats or 5 repeats or 7 repeats, you use something called gel electrophoresis. You actually put the DNA in these different wells, and then you run it, and you can see, remember something, larger fragments move more slowly, so they stay at the top, and smaller fragments move more quickly, so they're down at the bottom. And here's the genetic code. It turns out that when you see, let's just take one little look at it. Okay. I mentioned, like right here, that's TTG, right? Remember, it takes 3 base pairs to make an amino acid. And these are the different amino acids. You don't have to know them all. But this one, TTC, makes leucine. Okay? Some of these are redundant, but in general, you've got 20 amino acids. If you remember back from the molecules we were studying, 20 amino acids. Some of them we can make ourselves, and some of them are called essential amino acids. Those are about 7 of them. Okay. So, the DNA is wrapped up and are found in the chromosomes. Now, the only time you really see chromosomes is when a cell is about to replicate. Otherwise, it's kind of spread out all over the nucleus. There's over 3 billion base pairs long. The actual coding section of the genome is maybe 2%. So you might ask, well, what's all this other stuff? For a long time, it was considered junk DNA with things called, let's say, transposable elements. These are sections of DNA that are copying themselves. They're already in the genome, and they're copying themselves to other parts of the genome. They're kind of like parasitic DNA. And then there's some other stuff that was just thought to have no function whatsoever. And now we're starting to realize that, in fact, at least 20 and maybe more % of the genome is dedicated to gene regulation and gene expression, and not just for the coding region. In other words, genes that actually turn the coding region on and off, but not on and off, but more like a rheostat, a dimmer. So you turn it to the right or your right, that means more protein, something like that. So we're going to look at that. But first of all, we have to learn exactly what happens in terms of transcription and translation. Now, the DNA encodes, well, kind of a recipe, but remember, genes are always kind of in the context of the environment in which they're found. So, and DNA doesn't actually do anything. It has no, what you would call, well, it doesn't run any chemical reactions or anything like that. It's just like the letters in a book. And so it then has to be transcribed, and we'll look at what that means. Okay, so here's the chromosome, the gene. Remember, proteins are made of amino acids. The genetic code, which means a codon is 3 base pairs long, codes for 1 amino acid. And those are strung together in proteins. And you can see all the different okay. So why doesn't this work? There we go, maybe. Jesus Christ. Anyway, so this is actually the sequence. Okay. So here we have the DNA. Remember, you've got a complementary sequence in it, so the bottom one okay, T goes with A, C with G, all the way down the line. Now, at this point, what happens is the DNA actually unzips, and the RNA is actually an RNA. Now, an RNA is not double-stranded like the double helix. It's related to DNA, but it's not double-stranded. And so this genetic code is then translated into RNA. Now, you might see something here that's a little confusing. So up here we saw the T, right? Well, RNA has a different, it's called uracil. So instead of a T, it's actually a U. But it codes for the same thing, okay? So when you transcribed it from DNA into an RNA, all the T's turn into U's. Got that? Okay. And then the RNA is ferried out of the cell into the cytoplasm and then translated actually into a protein. So this is actually what it's like. In fact, I think I want to watch this little God damn it. Oh, for Christ's sakes. Fuck.
I think since it's a text, you can't really click on it.
What?
Are you trying to like click on it to go to the website or something?
Yeah.
It's a text. I don't think you can do that.
Okay. I'll just have to go to the website. Come on. Come on. You can do it.
Hold it now. Now hold it. There you go. And then see, it's just like oh.
Okay. Why is it going to... Oh, look up there. There we go. Oh, for Christ's sakes. Yeah, okay. So, this is the way things go. It goes from DNA through transcription into RNA. It's called messenger RNA, mRNA. And then translation in the ribosome, and it's actually, there's a transfer RNA that's used. And from there, it's, the amino acids are strung together in a protein. And then, actually, you can't see it in the cell, but there's some other structures in a eukaryotic cell that then modify the protein. So you can actually get more, like a bunch of different types of the same protein. So it always goes like this. DNA is transcribed into RNA and then translated into a protein from the RNA. I gotta sneeze. Damn, it's not gonna do it. Well, it'll happen. Okay. Okay, now, do the cells have only the genes they need, or do they have the whole genome? Right. So it's not like an eye cell only has genes for an eye. Okay, it means it's got the whole genome, and then the genes that are needed for an eye to function, first to develop and then to function, are then expressed. And this is called gene regulation and gene expression. And the gene expression in this case is the expression of a protein. And not just kind of like an on-off, it's more like an amount of a protein. So on the left, there's muscle cell. On the right, there's neurons, right? They look different. Now, it turns out they all use some genes all the time. These are housekeeping genes that are just for cellular function. And then in terms of the specific cells, they're certain genes are turned on. So, okay. Now, let's go on. So remember, sperm and egg, that's haploid. That means there's a copy from mom. Okay? Sperm, dad. And then a zygote has both. So we'll say this is N, and this is N, and then a zygote is 2N. Okay? That's why there's 23 pairs of chromosomes, and they turn into 46 total chromosomes. So there's a pair of chromosomes that are identical to each other in terms of the loci. The genes that are on it, they will have perhaps obviously different alleles at different sites, but there will be 46 corresponding to all the chromosomes. Okay, so here's a muscle cell. We've got gene A, gene B, C, D. This is the DNA. Transcribe the RNA, translate it into protein A. Same thing with B and with C. Now, the protein, each circle is the amount of protein, the number of molecules of the protein. So in this case, protein A, there's 1, 2, 3, 4, 5, 1, 2, 3, 25. Okay? Protein B, there's only 2 molecules, and protein C, there's 13. Gene D is not regulated or expressed for muscle cell. A neuron, it turns out we still have the same amount of protein A. That's probably a housekeeping gene. Gene B is not turned. C is and then D is translated and transcribed and translated. Okay? Now, each gene has DNA sequences that control the gene expression. This is what I mean by gene regulation. There are promoters and enhancers. Promoters are usually at the head of the DNA strand and actually turns a gene on or off. Okay? Enhancers can be anywhere, including on other chromosomes. So the promoter region or the enhancers do not act like an on-off switch. Okay? They act instead like a dimmer. There's a dimmer in here. Doesn't look like it. Okay. Anyway, you get the idea. So the more the dimmer is turned, the more protein is transcribed and translated. So the protein area, the thing that's actually being turned into a protein, that's the coding region. But as I mentioned, there's all this other regulatory stuff going on in the whole genome. You know, there's over 3 billion base pairs. And a lot of it has to do with gene expression. And it does it with things that are called transcription factors. They're actually small little proteins that then fit into certain slots in enhancers and promoters and get them to do their work. So this is from a recent thing in Nature. B, so there's different regulatory regions. So scientists can identify functional regions in the DNA that are active in modulating gene expression by combining results from biochemical assays with evolutionary comparisons between species. And so you have all these different species, and they share many of the same genes. The regulatory elements active in different tissues, red, brown, and green peaks, or common to all, are identified by open chromosome histone modification or a transcription factor. You learned a little bit about the histone protein and all that. So you don't have to know that, but that's how it actually is. You can see these peaks. Now, evolutionary conservation, these are the yellow peaks, reveal conservation aligned sequences from different species to reveal regulatory elements that are conserved and thus must have a role in gene expression. Now, a lot of the regulatory elements are actually located far from the genes that they control, but they can be matched. So you can actually, some of the molecular techniques we have now, we can actually determine which genes are regulating the expression of the protein. And then you have this network, compilation of all the regulatory element connections to their target genes builds a network of regulatory interactions that is unique for each tissue. Okay? So this is more than you need to know, but it's pretty amazing. For a long time, it was thought that a gene had a mutation, you have allele, and perhaps a new function. And so that's the way evolution works, right? We have genetic substitutions for base pairs, and you end up with different proteins, right? And that was it. But it would mean that evolutionary change would actually take a very long time, multiple generations. Well, it actually turns out that organisms are a lot more flexible than that. So organisms in different environments will often, the same organism in different environments will often express different proteins to deal with those. Now, sometimes they turn into substitutions, but a lot of times it's just a matter of which genes to express in different environments so that an organism can actually perhaps live. You know, a spotted owl might live in the Pacific Northwest in the redwood trees, but the Mexican spotted owl, closely related, has the same, most of the same genes. There's no trees in the Southwest. They nest on cliffs and things like that, and it's a much drier climate. Okay, so I think you should probably all watch this. It's about 15 minutes. It's pretty interesting, though. It's about lactase persistence. Now, remember, here we have, this is the lactase. That's an enzyme, right? And it breaks down lactose. And here's the lactose molecule. It's made up of a glucose and a galactose. They fit into certain substrates, and then they can break apart and turn that disaccharide into a monosaccharide, namely a glucose molecule, that can then be used, yeah.
I am like very lactose intolerant.
Yeah.
My whole life I've been drinking milk forcefully, even though it upsets my stomach because I don't care. Is it possible that I'm mutating that or no?
No, doesn't work that way.
No.
No, you've got what you got. Yeah, yeah, I know. See, for me, I wouldn't go that route. I would rather fly. And so if I would like to mutate my genes so that I could actually fly. But needing and wanting have nothing to do with whether you're dealt that hand or not. I'm sorry. Bummer. And you like milk too.
Yeah.
Yeah. Well, one thing I will tell you, most of the galactose or the lactose is actually in the whey part of the milk. So if you eat cheese, you usually are not lactose intolerant to that. It's been modified so much that you can actually digest that. Okay? And I think butter is the same sort of way. Okay, so now, in terms of lactose persistence, there was just a single mutation. So here's the gene, of course, and then the mutation, we'll say right here. The C was turned into a G. This is the promoter region, right? And it turns on the lactase gene and turns it off. Normally, there would just be, you have a young mammal, and they're finally weaned, and at that point, because there's no more lactose being produced by the mother, that gene is turned off, and it's not turned on again, because it's very unlikely that a young mammal will ever come across milk again on a regular basis. So that's why there's lactose intolerance. There's no reason to be producing a protein, in this case the enzyme lactase, which is expensive to make, when you don't need it. But it so happened that with humans, there was an evolutionary substitution in pastoralists who herd cattle, goats, sheep, and it turns out they'd probably try milk and get upset stomachs, but they really couldn't get much out of it. But there was one individual who had a mutation, and suddenly it kept the lactase gene on. Normally that would be a bad thing, but in this case it's good, because that individual can then drink milk and has a food source that no one else can actually access. So who's going to leave more offspring in the future? That individual, right? The population is then, that gene will then, that gene in the promoter region will then spread through the whole population, and you'll find a population composed almost exclusively of people who can actually digest lactose. Okay, and it's happened at a number of different times. It's happened in the Middle East. It's happened in Africa at least twice. Might have happened independently in Europe. I'm not sure. They're not sure about that. Now, RNA polymerase, that means that's another enzyme, right? And that's that whole machinery that we saw in the video. And what it does is it takes these RNA molecules and creates this long strand of mRNA. Here. So this is the transcript of the DNA. And remember, uracil U instead of T. So this is the way, this is called essential dogma. You can only go in one direction. You can only go from DNA to RNA and then to a protein. You cannot go from a protein and then back translate it all the way into DNA. So that leads to there's a lot of questions about how life actually began. First of all, it's probably that the DNA, the first cells didn't use DNA, but they used RNA. And DNA came along because it's more stable, and so there was a mutation, a double-stranded RNA that became a DNA. RNA is single-stranded, and so there's higher mutation rates on them. And then, as I mentioned before in the video, you end up this tRNA, translation RNA. The translation occurs on a multi-protein complex called a ribosome. This is the whole ribosome. Right here. The tRNAs are these, and they actually, what they do is they glom onto a specific amino acid. So here we have AAA. It codes for a certain amino acid. UAU, another one, UCC. Then there's actually something called a stop codon that tells the whole assembly to stop. That's the end of making that protein molecule. Okay. So here's the binding site. This is the amino acid that it's carrying. Okay. Phenylalanine. Each one of these transfer RNAs, which is this whole molecule here, okay, carries one specific amino acid. So there's all these tRNAs that are floating around that are then taken into this ribosome and turned into a long polypeptide of amino acids. Okay. Now remember, it's always a 3-letter code, so it's called a codon. This is the anticodon, by the way. So it's actually the equivalent of the complementary sequence. And then it binds the binding site for the amino acid. Okay. Now, remember, DNA only produces proteins. And they actually produce a whole suite of RNAs for regulation. But in the meantime, this is a simple example, right? So we have these amino acids for the taste receptors. And okay, so sour, salty and sweet, and then bitter. And there's actually another one called savory. And taste varies among people. Anybody in here like cilantro? Cilantro. Anybody in here hate it? Anybody? Yeah. Okay. What does it taste like? Soap or something, or what? What? Like dirt. Right. Okay. It's because she's got a substitution that makes cilantro taste lousy, whereas everybody else, a lot of people in here like it. Okay. These taste receptors, this is a matter of different alleles, right? So you can taste it, but you can't tolerate it.
That's the same for like being an alcoholic. If you drink it, you might love it?
Well, there's certainly a genetic component for alcohol. As far as whether part of it, of course, is learning, but obviously it kind of runs in families, right? Any Irish in here? Okay, I can. Oh, you are. Then I can't say anything. Anyway, certain groups tend to be more likely to be alcoholics than others. So it certainly runs in families. So this is a good example of gene expression and what you can taste. And obviously tasting is very important. Babies, once they start eating solid food, they're actually training their taste receptors on different things. So they often really don't like a lot of stuff because they've never tasted it before. I remember my sister, she hated everything, which was great because there was more food for me. And like one thing she hated was actually chocolate ice cream. I mean, can you imagine? She'd go, right? Make a terrible face. And the only thing she would finally eat was Chef Boyardee SpaghettiOs. That was it. Okay, obviously she needed vitamins, okay, and she wasn't getting any protein. Well, one day she was out in the garden and she discovered escargots, which are garden snails. And of course, they're not, instead of cooking them nicely in butter and garlic, like the French do, or civilized, she just started eating them, shell and all. And my mom looks out, and there she is eating away. And my mom runs out to stop her, and then stops herself and says, hold it. This is protein. She needs protein. And she let her just keep eating. Yeah, no, I know, but it's like, right. And that's the first time I tried to poison my sister, because after that I started sprinkling Snarl all over the place. Okay? Unfortunately, it didn't work. Okay? Anyway. Now, hemoglobin is a molecule that carries the oxygen. Most hemoglobin that you have is in your red blood cells. And here's a red blood cell. Now, this is a sickle cell. That means there's been a substitution that is not such a good thing. And you can actually see, let's see, for 1 second. We'll get onto this more later. But it turns out that people with one copy of the normal allele, HBA, and the HBS allele are resistant to malaria parasite. It tweaks the hemoglobin just enough that the parasite can't get into the red blood cell. Okay? So those individuals are less likely in a zone where there's lots of malaria to actually suffer from it. Now, the wild type, if you're just a regular type of hemoglobin, those individuals get really sick. A lot of them die, especially children, right? So there's really strong selection where there's lots of malaria. And malaria is a vector-borne disease, right? It's carried by a mosquito from one person to another. Now, it also, it's interesting, the parasite actually affects the mosquito as well, so that it preferentially bites someone who actually has malaria. You say, how does it do that? Well, there's different people smell differently if they have malaria or not. And it gets into the brain of the mosquito, and actually the mosquito can then discover that the person smells a certain way and goes and bites it. Okay, now, if you have one copy of HbA and one of HbS, you're resistant. Unfortunately, if you have two copies of HbS, this is a recessive deleterious trait. Those individuals are in bad shape. They get sickle cell anemia, namely sickling like that. These don't go through the capillaries as well. They start building up in certain places, terrible pain and all that. They're anemic, obviously. They don't carry oxygen very well. And so most of those individuals have like zero fitness. So that's a pretty high price to pay, but in an area where malaria is killing or sickening, 90% of the population, without the substitution, then it actually is worthwhile. Okay, genetically modified organisms. People have freaked out about GMOs. Of course, there's a lot of markets and stuff saying no GMO. They're not, they don't sell GMOs. GMOs are bad. This corn right there. Let's say that's a genetically modified corn. Tastes just like any other corn. Eating it, you'll get the same nutrients as you would from any other corn. Genetically modified organisms, in this case the corn, let's say, or the goat, they're affecting evolution. They're actually giving certain organisms new genes and new alleles that they didn't have. But it's kind of like selective breeding, except that with selective breeding, you're breeding for traits that usually correlate with a whole bunch of others. So you get a suite of traits that also change, whereas with genetically modified organisms, you don't. Okay? Oh, here, let's review. So, which cell type in your body contains the gene that encodes for your photoreceptors in your eyes? A. That's in the retina, right? Well, hold it. All your genes are found in the cell type. Remember, every cell has the whole genome. It's just a matter of which ones are expressed. So it's not A, okay? It's actually all of these. The whole genome is present in each cell type. It's only certain genes are expressed in certain cell types and not others. What is the name for the dimmer switch that regulates gene expression? Anybody? Starts with a P. Promoter. Okay. What are the two main steps that lead from gene to protein? Okay, good. Okay. If we insert a human gene into a bacterial cell, the correct protein won't be made because the genetic code is different between bacteria and humans. True or false?
False.
Okay, I have here one false. Any true? It's false. We share many of the same genes as a bacteria does. The same exact genetic code. In fact, which is actually pretty cool because then we can get bacteria, we put a gene into them, to actually make certain proteins that, let's say, we might need, like insulin or something like that. So, in other words, the genetic code is universal. It's obviously great evidence that show that we all share our most recent common ancestor. We do. Yay, science. Okay. Now, the technology that we use is called recombinant DNA technology. And before that, we actually got insulin from horses. We got horse insulin. It was isolated from horse blood and given to diabetics. Okay. Now, some people had a bad reaction to horse insulin, so they were in real trouble if they were diabetics. But now the human insulin gene is actually inserted into a bacterial DNA, and so you get a whole big vat of bacteria actually making human insulin instead of the corresponding horse insulin, and you don't get adverse reactions. Okay, first of all, it's a lot easier, much less expensive, better results for patients, much happier horses, because they get the DNA by bleeding the horses and using the blood, probably then putting it back in, but taking the insulin out. Okay, so these insulin-producing bacteria are called transgenic. And what that means is you take the DNA from one organism and you put it into another that doesn't have that gene or those alleles. Okay? So that's a transgenic organism. Now, the insulin protein that is isolated from the bacteria is called recombinant insulin, even though the actual protein itself is indistinguishable from the human-derived protein because the bacteria are making it. So they use what's called recombinant technology, where they actually substitute one gene or insert a gene into an organism. Okay, so can you think of anything else that you might get transgenic that might produce one thing or another that they normally wouldn't? Anybody? Take a look. So antithrombin is a protein that helps prevent blood clots. There are some people who are born with a defective copy of antithrombin gene, and it is expressed in cells in the liver, right, which release it. But if that person is born with a defective copy, they're not making it. So they have a problem with blood clotting. Okay. Now, before scientists, they took the antithrombin from people who were donating blood, and they collected it, and then gave it to people who were deficient. But it turns out transgenic goats can produce massive amounts of the protein in a relatively short period of time. So, transgenic organisms, in other words, where you have an organism with a gene that comes from another species, are called genetically modified organisms, or transgenic, right? Where do you think is a good place to have human anthrombin expressed in a goat? Like in the bloodstream, in the muscle tissue, in the milk? In the milk. You can actually have it expressed in the milk. So, what you do is you create what's called a hybrid gene. You actually use the goat promoter gene because it's basically the same promoter that we have. And then you attach it, you make kind of a hybrid gene that makes antithrombin. And then you go and milk the goat, and then through a process get all the antithrombin that you need. So what happens is you've got this thing where you have a microinjection and embryo transfer. And you take out, you have a cell, you take out the old nucleus, or the goat nucleus. Let's see. The hybrid gene is injected into fertilized goat embryos, right? These embryos are then implanted into surrogate goat mothers who give birth to these transgenic offspring. And then, of course, you purify it. Now, we do these transgenic things for all sorts of different things. It turns out there's this fluorescent protein of jellyfish that live in the deep ocean where there's no light. And so they fluoresce. They are luminescent organisms. We've actually taken the gene for this, and we've tagged it into molecules in a cell, and you can actually see how things are, the pathway for like gene expression and things like that. So it's actually been very, very useful in looking at intracellular activities that are going on that you couldn't follow before. Okay, so obviously they're used for medicine, research, agriculture. You can engineer bacteria to produce growth hormone for cows, engineer plants to produce proteins that make them more marketable, or perhaps that are resistant to herbicides, which is one of the big things now. Okay. So it turns out that bovine growth hormone, if you give it to cows, if you inject it, it makes them make a lot more milk. And so how do you get the cows to make more of this BGH protein? Well, bacteria can, of course, serve as factories for producing lots of BGH that you can then inject into the cow. Okay? So here's a question. Do the cows in this example qualify as genetically modified? No. Which organism actually does qualify? Where's this stuff produced? In a bacteria. So the bacteria is the genetically modified organism, not the cow. The cow is just getting this extra product, like someone with antithrombin deficiency would get the antithrombin product as well. Okay. So, now, some people will probably say, oh, they're freaked out about BGH. I don't know what effect it might have. And I don't know how long it's expressed for in the cow, so it's hard to say whether it's good or bad for you. They probably use it, you know, in terms of milk production, because you have a cow that makes a lot more milk. I'd say I'd be more concerned about drug-resistant bacteria, because they're giving cows and a lot of other livestock all these antibiotics. Makes them grow faster, but it also creates a lot of resistant microbes that people could then get. Okay. Now, we saw this before. This is called teosinte, and it was first collected and then grown in the highlands of Mexico, Oaxaca and that area. And over time, through artificial selection, in other words, farmers would pick which corn to plant. Some with the bigger kernels they'd plant more of, and eventually over time you ended up with a nice big stalk of corn. That took obviously many generations. The first evidence was like from 10,000 years ago. So we've already talked about selective breeding, but nowadays we can actually modify genetically modified corn or wheat or something like that, and there's a big controversy. Is that good for you? Is it bad for you? Genetically modified corn is actually no different than regular corn as far as the nutrients it has. Now, most of the genetically modified corn is not the kind of corn that you actually eat. It's the kind of corn that we feed to cattle to fatten them up. There's this genetically modified rice called golden rice, and it's been genetically engineered to produce beta carotene, which is vitamin A. A lot of people who are poor people don't get enough beta carotene in their diet, and so they suffer from beta carotene deficiency. So they've started to grow this modified rice in Southeast Asia and places like that. But, you know, there's been some people who say, well, we don't want it because it's genetically modified. Well, in this case, it's kind of I think it's kind of a dumb sort of thing to do, but each for their own. Now you can actually, by the way, you can modify plants with a gene gun. So you just basically shoot a new nucleus into a plant. Now, this is actually really old. There's a brand new technology, and it's called CRISPR, and then FA16. Okay, and CRISPR-Cas is a method where you can actually go in and you can cut DNA at a very specific spot, exactly where you want to cut the DNA, and then insert a stretch of DNA. Now, before, with genetically modified organisms, sometimes it would work and sometimes it wouldn't, because sometimes you wouldn't get it into the right place. With CRISPR-Cas, you can. Now, it's brand new technology. It was first discovered because it's similar to a process where bacteria have immunity to certain viruses. There are viruses called phages that infect bacteria. And CRISPR-Cas is a system that actually is kind of like an immune memory for sequences of viruses, and when the bacteria find that sequence, the CRISPR-Cas system goes into action and starts cutting up the bacteria. Well, people discovered this, and then someone thought about turning it into something that we could use. And it's an extremely powerful technology. It's a little scary, actually, because before, a lot of genetic modification was kind of a hit and miss sort of thing, and CRISPR-Cas is not. It's exact. There's a big debate on who discovered it, but actually there's no debate. There was this French woman and this other woman up at University of California, Davis. And they got together and they actually developed the CRISPR-Cas system. And they're probably going to win a Nobel Prize in medicine. Okay. Now, the thing is, you can also do a lot of things that you may not want to do. We'll call it there, and then on Monday, we'll finish up, and then we'll go, if you want, we can go over what's going to be on the test. Okay?